Okay, you're all welcome to today's. Okay, um, well, welcome once again. Yesterday, in our previous class, we were able to um, carry out the docking process from scratch using Autodoc Vina. Um, we were also able to perform the required post-docking analysis. We call that post-docking analysis, which involved visualization of your docked results in their 2D views and 3D views. Okay. So we were able to carry that out yesterday, and then today we just want to perform one other or two other functions, activities with respect to our compounds of interest. Mhm. Yeah, when investigating a compound for its drug candidacy, aside docking the compound against the target, you also try to carry out one or two studies, with respect to the compound, um, to further establish the drug candidacy of such compound. Yeah, part of what we'll be looking at firstly today is the ADMET analysis of compounds. Yeah, the ADMET analysis and drug likeness properties of compounds, okay, which are our potential drugs and all that. So we want to see what are the properties of these compounds or drug candidates we just docked that probably had good binding scores or pools, after docking. These compounds, they fit, for drug development, considering their pharmacokinetics properties. When you talk about ADMET is absorption, metabolism, uh, yeah, distribution, absorption, distribution, absorption, and toxicity. Okay, that is ADMET. Absorption, uh, distribution, metabolism, excretion, and what toxicity. With respect to these compounds of interest, how are they absorbed when consumed? How are they distributed within the system? How are they metabolized? After that, how are they excreted? And then what's their toxicity profile and all that? Is that okay? So there are quite many servers or tools, excretion. Okay, so there are many servers you can use for such functions. Okay, for the course of this, we're going to be using the Swiss admin. Okay, one thing about working with some of these is whichever tool or server you're using, make sure it's a well recognized server. Okay, yeah, that people know about. And then when you're publishing that, it will also be allowed as well. Okay, so you don't use just any kind of server that at the end um, might be questionable for its authenticity. Okay, so to do that, you simply type Swiss admin. Okay, admin, Swiss what admin. If you typed it correctly, this is the icon. So you simply hit on your enter button to open that. And then when you open Swiss admin, you're going to find in the first page, you're going to see Swiss admin as the first one here. So you simply click on Swiss admin, and then this is the page. This is the home page. Okay. In the home page, you can see these are other servers, by this particular, um, institute, okay, those that host this. This is just Switzerland, as you can see. This is drug. This is used to estimate or predict drug features and properties and all that. So the same with some of all these other ones here. Okay, so when you scroll down a bit, you discover there are two boxes here. Alright. The first box is where you could draw your compound, assuming you don't know the structure or you don't have the structure already. This comes into play for compounds that you synthesized newly. Is that alright? You know, if you synthesize a compound newly and then draw it with ChemDraw and then possibly um, converted to 3D and work with, you might possibly not find that in your PubChem, right? Yeah, so you should have to find a way, of getting some of these properties. It's only those in the PubChem already that you can simply pick. You can see here in the second box, you can see you can search or investigate, predict the property of any compound using the SMILE number. Okay, we call this the canonical SMILE. Each compound you open in the PubChem, if you looked very well, you see there is a section where you have the canonical SMILE number, you have the alpha name and all that. So for compound that you synthesized or you drew yourself, which you didn't download from the PubChem server, you won't have the canonical SMILE there, right? So you will have to paste the 2D view here. Do you understand? The same compound, the same way you drew it in ChemDraw, you simply copy it and paste here instead of coming here to draw. Yeah, to start drawing. Is that alright? Yeah, yeah, in 2D. The same way you after drawing the compound, it appears in 2D. Alright? Yeah, before you now convert to 3D. So you simply paste it here and then once you paste that, you can now click your run. This run will become highlighted. However, for the course of this, we are going to make use of, we have been working with naproxen, right? Okay, so yeah, we don't need this because we have our own. Kianfiro 2 is in the Chem server. So what do? I'll go to PubChem. I'll go to PubChem. Okay, I will search for naproxen, okay, which we started working with since yesterday. So I'll simply search for that. Okay, and then naproxen has appeared here. Okay, so if are you with me? Yeah, even at this point, you can see what the canonical SMILE or this isomeric SMILE, you can see it here. Okay, you can find it here. If you open the compound as well, assuming I click on the compound and it loads. If I scroll down, okay, if I scroll down, I'm still going to find under these names and identifiers, you are going to see the pack name. Is that alright? Then if I go down a bit, this is an Inkai name as well. This one is Inkai key and all that. You see the what? The canonical SMILE. Is that alright? It's the same as with the isomeric SMILE. Okay. It understands the both of them. Okay. So I'll simply copy the canonical SMILE. I'll copy that. Is that alright? Copy that. Copied it. Then I'll go to this wizard mail, right? Then I'll paste it. Okay. Let me show us what will happen. Okay. This run is not highlighted here, right? There's nothing you can paste. You can click there. If I paste this will get highlighted, right? Meaning it has recognized something inside there, so you can actually run it. Is that alright? So I'll hit on run. Okay, and then it has run. Is that right? It has run. And then, of course, this is our compound. If you can still remember the structure, this is our compound. And then, um, this is the phytochemical properties. Okay. If you start reading or going through article, you discover that some people, even have a way of just snapping the entire of this box here, you know, and paste in their publication article. Okay, just to show the totality of the properties of that particular compound. However, um, snapping all might not be too necessary. So what we usually do is we get a table, okay, where we pick few important subheadings and then points from this particular table. For instance, if you go to the phytochemical properties, the molecular weight is fine. It's very, very important. Okay. If you wish, you could add the molecular formula. Okay, the molecular weight is fine. Then the number of hydrogen atom, uh, number of rotatable bonds, acceptor and donor is very, very important. You might not need this rotatable bond and all this. If you still remember three, when we were about docking, when we checked number of the, do you remember it was three there? Okay, that torsion three, yeah, that was three. Is this still the same thing is telling us here, which is this three under here. Okay, then after that we have the number of hydrogen bond donor and acceptor. They are very, very important. Okay, they are product of how our compound could react. And then the molar refractive index is also important here for this phytochemical, under this heading, phytochemical property. Under lipophilicity, okay, what we usually pick out here is the consensus log p value. Okay, I'm going to show us what this and all that comprises later. Then under solubility, is it soluble or non-soluble or very soluble or poorly soluble? As you can see, this is just soluble. Okay, the same when you come to pharmacokinetics section, there's what we call GI absorption. Okay, yeah, that's intestinal absorption. Is it high or is it low? As you can see, it's high here. If you hover this your mouse around this thing, it will help you to understand what it actually means. If you hover around this thing, for instance, this Lipinski, if I hover my mouse around this arrow, can you see this question mark? Yeah. So it's telling us what this Lipinski actually means. Lipinski is a rule that depicts the drug likeness property of a particular compound. Okay, there's what we call the Lipinski rule of five that states that if I hover this, you discover that row. This is one, two, three, four, and then the fifth one, which is now here. The C log P value is not okay. M log P value is here. Molecular weight is here. Um, okay, I'll get the one that is not here later. We have 5 rules of the Lipinski. You can as well read about that properly later. The first one is that the molecular weight of the compound or the drug candidate must not be greater than 500, right? It must be less than or equal to 500. You also have that the M log P value, which is this consensus log P value here, must be what? Less than or equal to 4.15 for an ideal drug. Also, the number of hydrogen donor, okay, which is this one, hydrogen acceptor. Sorry, the first one is hydrogen acceptor. This end that doesn't have hydrogen is the one accepting hydrogen, while the end that has hydrogen is the one that is donating hydrogen. Is that fine? So the number of hydrogen bond acceptor or number of oxygen donor, is that right? Must not be greater than 10. As you can see here, this one is 3, so it's fine. All right. Then, lastly, the number of hydrogen bond um, as donor, okay, hydrogen bond donor must not be what greater than 5. Okay, If the compound obeyed all these rules, you discover that the Lipinski rule was obeyed. There was zero violation. All right. However, when you start working with different compounds, you might discover some compounds are quite complex, something like alkables, protein, and all that, which are complex ligands. You discover their molecular weights are quite high. You understand? In such case, you don't classify them as bad drug. Rather, you that will affect their dosages and all that. Is that fine? That will settle the case. Okay, and then
So there is a, if you run some this place can show no.
yes, yes, yes. If
That means
the
it can
molecular
obey the
weight,
rule.
if the molecular weight is greater
Greater
than
than.
500, for instance, you see something like no, so and so violation. They will state the violation.
So if it is just one that disobeyed, will they still put no?
Yes, you still put no and highlight the violation that was there. Is that alright? You simply copy it. Let me show us um, an example of the table that we can use to compute our this. Um, Doctor, let me check doctors. Um, okay, let me see if I have. Okay, no, it's not here. Let me go to my downloads. I want to check for tomorrow. Tomorrow results. Just trying to recall.
All the money.
Oh dear.
All the money they have charged.
Tomorrow day. Okay, let me check here. Where is this file? So, okay, Benin.
Now baby now get us about. Tell you that all of them go to
Dr. Ben Pharmacokinetics. Okay, let me see. Okay,
can't table with this.
okay, so this is one of the table. You're shouting Jesus. Okay, so this is it. I worked with over 18 compounds or so with their standard here. Should even be more than 18. So this is 1, 2, 3, 4, 5, 6, 7, 8. 8. Okay, with this. Okay, so roughly that.
Think I am for
Yeah, yeah. Did this? Yeah, just increase the size. So these are the compounds. Can you see that? These are the compounds. So you can see naringenin, epicatechin, lunamarin, naringenin, reversterol, catechin. This is camphorol, the one you'll be working with. Is that fine? So you can see that maybe I can send this to us later to study carefully. You discover that there is pharmacokinetics properties or phytochemical properties. We saw that there. So these are what you can feel here. You can see their molecular formula. Then you can see their molecular weight. You can see number of hydrogen bond donor, acceptor. Then you can see that of donor. You can see the molar refractive index is there. You can now see the nucleophilicity, which is the seal of P value. You come across that too, right? And then the water solubility. You can see that some are soluble, some are moderately soluble. You can still see very soluble. Okay, then under drug likeness, okay, of the same compounds, do they obey the Pinsky rule? You can see that obeys the Pinsky rule. The first one is yes, 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 zero violation. However, this one is no. You have what, three violations. Okay, the molecular weight is greater than 500. Number of hydrogen acceptor is greater than 10. Hydrogen donor is greater than 5. And you can simply find that from here. The molecular weight of this is 5. The molecular weight of this is 580. Can we see that? The donor, the acceptor is over 14. The donor is over what? 8. You get that? So that is how um, this table looks. So you have drug likeness property, you have pharmacokinetics, okay, which are still there if we remember that. Under pharmacokinetics, this is what they mean here. Okay. So you can see there are some that somebody was asking if you can have load there. Of course, these large compounds that are higher in molecular weight and all that, their GI absorption as well. So yeah, it's quite slow because of their weight and all that.
About wait.
Yes.
What if we synthesize compound after running it? Will it show all those properties under them? Like the one we have.
Yes, once you drew the compound up there and run, yes. Except that you won't find the canonical smile.
So you run straight.
Yeah, you just draw, yeah, and run straight.
And that's it. Since we copy this state from somewhere, let me say you're writing a paper. You have to cite where you copied it.
What exactly did you copy now?
We got it from
The canonical smile.
uh,
Are you sure it's also
not that one. We're talking about
this table?
the properties that you copied from that Swiss admin.
Okay, yeah, certainly. You write down that in your methodology, the pharmacokinetics and ADMET analysis of the studied compound were performed using Swiss ADMET. Okay, in your reference, you still reference the Swiss ADMET there. Yeah, it has a reference. You have author. Same way. Yes.
Yes.
Yes. If you have also seen papers where it's just that when you're looking for, you have seen the paper, right? Yeah, I have many of them here, but if you start looking for some works like this, you won't be seeing them. But I know that all the people I've worked with, you write your methodology for them, yeah, and then their result. In the methodology, you will include all the tools you used. You understand? Include all the tools you used, and then after including that, in your reference as well, you list their attached references. Okay, it's there. I can share that with us later and all that. So this is how you get your table. Yeah, for each of the compound, as you can see, this is over 10 of them. All the compound added with the standard drugs I worked with. This quinolin trisulfonamide, this is galantamine, this is prednisolone, and this is acarbose. Okay, this is what I was showing us before. This is acarbose. You find out that it almost disobeyed all the rules because of the structure.
Oh yeah, where is that binding now? This is the result of it.
Yes.
That we got from.
Okay. Okay. Okay.
So that means any drug that contains alcohol is like the dosage. Because you said there is not a bad drug.
Yes, it's
It's
not.
just that the
Acarbose
dosage will be
is the most recognized anti-diabetic drug. Okay. Yes. For both clinical studies and otherwise, if you're administering to rat, this is what you use as standard drug. Or maybe later, but acarbose is the universally used one. Then she was asking how come we don't have the binding scores here. Okay, yes, like I said, this particular study is on the pharmacokinetics and drug likeness properties. Pharmacokinetics is always the last result after the other results. Your 2D, your 3D, they are not together. This is just a further insight into the compound. They should certify those other ones before you could now study this. Is that all right? Okay, so this is what we have, and then at the end, you will group all this the same way it is like this and forward to them, and then it will be attached in the document. Is that fine?
So this thing will be in your work, your paper.
Yes, yes.
The
However,
main work will be appendix.
in the main work, however, all these things has meaning, right?
Yes.
All these things has meaning.
So you discuss it.
Yeah, you certainly interpret this. However, you don't interpret one. When you start reading documents or articles where this was done, you see how the base interpreted. For instance, the bioavailability score, okay, which must not be less than 0.25. Okay, bioavailability score, which must not be less than 0.25. Okay, tells you how available or how absorbed is a product of absorption or absorptivity, how these compounds could be absorbed when administered. So if the score is less than 0.25, it means they are hardly absorbed. As a result, they won't be available internally. They won't be bioavailable. This is one of just an example of what the interpretation of this table will do. As you can see, it's only this, and it's always those compounds that are quite high in molecular weight. Something like naringenin here is having that. Something like acarbose is likely going to have similar challenge again. Okay, you understand that? Yeah, because of the structure, the absorptivity as well reduces. But despite that minute quantity that was absorbed, they could be very effective and all that. Is that all right? Okay, so that will be that for this. Um, the next thing I would like to show us also. Okay,
Is it just for the nitrogen that you look at the
you look at all the compounds you've studied, all the compounds you docked. As you can see from that table, there are many compounds there. But I only use naproxen because that's what I want to eat for the course of this study.
people don't understand this. People don't understand this thing. Let's say you are working with a plant now. Maybe that, like what you told me, that we have like 16 this thing.
Yes.
So in that one of my this thing, I will have 16 different. You said the list is 20.
Yes.
That is how your this thing is. Not just one. We are working with just one.
I say like for you now, yeah, you might decide to just make your own 20.
20.
Yes. Then out of that,
Can't I do less than that?
yes, you can. You can work with 20 and now do kind of screening. Your first study will be to screen.
So we
If
are
at
going
the
to be
end,
talking for everything, like
yes, normally, if you characterize your compound, you do what we call screening by talking all the compounds and now sieve out maybe best 10 or best 5 out of over 50 of them.
how will you know the best 10 of best?
Yeah, from their scores. Yeah, the ones that give the high top scoring ones, we call them
From
top
the
scoring.
binding affinity.
From their binding affinities. You are not supposed to characterize, do 2D and 3D of all of them, and then do pharmacokinetics of all of them. Do that with respect to. No, good morning. And good afternoon. Oh, no, they just took it. Okay, so that is what you have. But you certainly do this for all those top scoring. You are going to study or carry out the study for all of them. Those ones you characterize.
How many years will it take me for me to do that?
It doesn't take time. Okay, so no, it's just for like okay. So that being said, let me show us as well.
So, excuse me, sir. The three this thing that we did just now, won't you save it? Or you
Of
write it
course,
down or you copy it?
because you already have a document where you are filling. Is that
You just be copying and
that document that you're going to save later? Okay, you can okay, so quickly. So all these compounds, we are talking all of them. Yes, you can have more than one protein.
go with the 3D.
You can have uh, you can have more than one uh, protein. You understand? Whichever protein you have, you can dock as many compounds as possible against them. Is that right?
So this naproxen now is a standard drug.
Yes. For
Be using that one standard drug.
naproxen is a standard drug. Okay. Which was once a phyto compound before it was now optimized, and any drug you come across was actually a phytonutrient initially before it got developed into a drug. Is that not so?
Okay, so whatever you are doing is against the standard drug.
A standard drug is just a comparison, a reference point. You dock your compound, you compare your results with the standard
If
drawing.
your own is better than the
Yes.
standard, or if
I know say na if you have nine of that compound is comparison with that same standard one.
Yes.
that one is better than
Okay, so we are done with drug likeness property estimation or prediction, and then we are moving on to this other aspect before we move on to the next as well. I mentioned when you synthesize the compound, right? And then you needed to perform docking with that particular compound. Uh, you won't find that compound in database because it's yet to be uploaded there. So what do? You will draw the compound in your chem draw, okay, and then convert
This
to
thing is software.
yes, this is chem draw professional. I just opened them from my system now. All right, this is chem draw. I will install that afterwards to you people. Um, and then chem 3D is in this office or in the suite. If you simply go to your window button and go to all program, okay, and then you scroll down to chem draw, you will see chem office. My own is 2021 version. Okay, if you click on this arrow, you are supposed to see all that is inside this. Is that not so? That's where you might find your own if you have installed it before. You are going to see chem 3D there, chem draw, even chem script and all that. All right. So I've opened it to you. This chem draw and then this chem 3D. Okay, I want to draw the compound. I have two options here. I can either draw the compound individually by clicking on this and then drawing one after the other. Okay, yes, drawing one after the other is what you can have and all that depending on what you are actually drawing. All right.
Look for the structure online.
So, which one?
How will you know what you are drawing?
Yes, after synthesizing the drug,
Mm.
you will characterize the drug. The characterization, we don't call it drug though. After synthesizing the product, you will send the product to NMR for characterization. The NMR will detect from what is the possible structure of the compound. It will give you the 2D structure of that compound.
Okay, so it's from there you will now
Yes, it's from there you can actually copy
want to
it.
know what you are going to draw. If you
Okay.
are getting it. Okay, you can copy from that.
I don't know how to do it.
It's not here, we are not doing characterization.
we supposed to do NMR in my work self.
So in that case, is assuming you make up your mind to buy one for us, you buy, I think
I'll keep it in Nigeria.
30 to 50 million can buy one. Okay.
It's not even that expensive. So why is it that nobody is providing
I'm
that
just
in Nigeria?
telling you so that some
you are
people
not
in
in
chemistry here, these last people that graduated from chemistry here, some of their supervisors sent their students to us with they actually I think they sent theirs to South Africa through
Our
DHL.
own couldn't
I
do
have a
it.
friend that even did one.
Just
So
in case you are not in the country.
even if you're in the country, some of them from chemistry can
Yes.
contact you. I've worked
No, if you
some
say
of
now
them.
what is the purpose of this thing when we are not even doing NMR
Yes, you
It's very, very important to us.
yeah, you in particular might not do it, but if you are good in docking, people that did that might meet you
okay.
with the name of the compound and maybe the 2D structure of the compound and say we want to investigate the therapeutic effect of this our synthesized compound. Do you understand it? So you will not tell them, I don't know any man. No, you just pick their compound. That's your own consent and now start working with. You got it, right? So if you have the name of the compound or the 2D structure, you can copy it. Normally what they send to them is the name. Alright? Then they will now paste the name here and then generate the structure. The name will come as canonical an IOPAC name. Alright? Yeah, these two
So you
understand.
paste it in chemical.
Yes. You can use
What if you paste it in that first interface of SweetMed?
that one is to estimate your pharmacokinetics, whatever. You don't use that one to after drawing them you save. No. That one is if you paste anything there is to estimate the properties, to predict the pharmacokinetics. Okay, so what do? You will open the software. Yeah, this is an offline tool. Okay, it's an offline. Is that alright? So you will go to, if you have the name of the compound without the structure, you will go to this top option where you have structure. Is that fine? You click on structure and then you will scroll down. Your own system, everything might show, but mine, I might need to use this arrow to go down. Part of what I will see down here is what? Convert name to structure. Alright? So convert name to what? To structure. You can see that most of these are what? Unhighlighted, right? Okay. Okay, let's continue. So I was saying to, if you have the name of the compound, the sized product, you simply come to your structure. Okay.
According to what you said, say NMR will detect the name.
Yes, it will
According to you.
identify the name.
You just come here
Yes, to draw the
to
name.
structure, convert name to structure.
Okay, so you hit on this convert name to structure and you paste the name here. So assuming the name is now Proxen, which is what we'll be working with, right? Okay. If you leave this highlighted, it will paste it and then it will give you the name. Okay, you can see this highlighted, but I might not need. Okay, let me just highlight that. So I'll click on that. This is now Proxen. All right. If you remember the compound, this is actually how it looks. All right. So if I don't need this, I can take this off. So this is
So
the
if you
compound.
unhighlight, what will happen?
If you
Unhighlight that thing. That
this name will not come out. It will not paste the name. Okay. So what do I do? This is the 2D diagram of the compound. However, what I'm working with is 3D, right? So I'll simply copy this. Okay. I'll do what? I'll copy it. To copy it, these two first tools up here, if you click on them, you see this and this. They are both for highlighting. All right. They are both for highlighting. This one is when you're highlighting things that are not straight. All right.
the first one.
Use the first one. However, if you're using the second one, you could actually highlight this. Is that fine? So if you highlight, you can copy this by doing Control C, or you can right click and copy. Is that right? Okay, I've copied this. So I'll go to Chem 3D. All right. This is Chem 3D. I'll simply paste here or Control V. All right. So I'll do Control V, and then it has pasted. This is it pasting. This is it pasting in what? In 3D form. Look at the 2D here. Alright? This is the 2D.
Excuse me. The place you got this one is just the chem drop.
Yes.
Then
This is an extension of ChemDrop. ChemDrop Professional is the suite.
chem 3D.
This is Chem 3D. So I've pasted it here. I'll have to save this out here as a PDB file. It's now in 3D, but still not in PDB file, right? So I'll have to go here and then I'll go to what? Save as. Alright? I'll go to save as. I can see that here. Okay, I've actually selected it before, but if you've not selected it before, you might have a different extension there. So I'm supposed to look for what is it called? Routing data bank. Okay, and pick. So in case you do yours and this is not there by default, you have to select it. But when you work with it over and over, it will appear like that. Is that fine? So I can now save this as map proxen. Alright. And then of course I'll navigate. This is a different folder. Now I'll have to go to tutorial classwork and then I will change to complex one because I touched that. Is that right? Okay. So this is my proxen. I'll do what? I'll save it. It will tell you this. You just leave that. It's already saved. Is that fine? So I can now go to that folder and then I have the compound there. Okay, look at the compound here. Okay, that's exactly this. Is that fine? So I can now open this and start working with it. taking. Do we have any confusion with respect to this? Okay, then,
Excuse me, you were drawing something before I interrupted and said, how will you know what we are drawing? So if you actually want to draw those things yourself instead of going to structure.
okay, yes, if you want to draw those ones yourself, you can use these tools. All these here are for your drawing. This is single bond, this is double bond. Then you see this, which is
Maybe I should illustrate. Let me see.
yes, you can pick. Okay, let's say this is benzene ring. Okay, this is benzene. And then, assuming I want to, this is a cyclohexane, right? Not benzene. Okay, look at benzene here. So this is cyclohexane. I want to put a double bond here. I'll click on this double bond here, and I'll come this portion and put double bond here. I need another one here, and then I need another one. All right. If you mistakenly put another one here, what do you think will happen? Look, there will be an error. All right. The benzene may not be able to because of that. Where you will notice that is assuming you put something like this. Okay. You see the red this thing is telling you that it can be more than 4 bonds to a carbon. All right. So this is how you draw. So any of these two that you need, assuming I need a single bond to connect this, I'll simply draw this way. I need another benzene ring. I simply draw this way. Make sure you are touching this end here before you could draw. Is that right? Okay, so that is how. So I can remove this. Okay, so this is what we have, and then this is what we have. Okay, then if I highlight this now and go to the same structure, you discover that many of these have become highlighted, right? uh Some of them, even to predict the NMR, whatever, yeah, is something. Then here you can see convert structure to name. You now have structure, but you don't have the name. Okay, you can as well do that. This is the structure to name. Let's see what it will generate for us. You see that this is actually the impact name. Of course, you might not recognize it as naproxen, alright? It will give you the chemical name for that. So that is fine, right? Okay, quickly let's go to um the remaining thing we have
Let me check if the power bank is dead.
before we round it off. Okay, the next thing I will show us is okay, let's pay attention. The next thing I will show us, I mentioned that earlier while we were trying to give us a few things we will be considering. In cases where you want to perform an analytical study and then your protein wasn't found in the database. I think I mentioned something like that.
Yes.
Okay. Yeah, you have this protein in mind you want to dock with, and then you check the PDB. The structure isn't there. All right. Yeah. Possibly maybe because the protein came out new, or the protein hasn't been elucidated structurally before then. So what do? You model a protein. Okay. You model a protein for that particular study. A model is a kind of a similar copy, okay, of your protein. By having similarity with your protein of interest, it should exhibit similar function. Okay, there's this sequence structure function relationship in protein chemistry or in molecular biology as a whole. Is that right? So if you have similar sequence, you should have similar structure. Similar structure should exhibit similar function. Alright? So while modeling, you use the sequence of the once a protein is identified, the next thing they do is to sequence it. Alright? The protein can come out today and before 1 week the sequences are all over. Alright? Yeah, but the structure might take a little time, weeks before it could be elucidated. So when you have a protein structure or a protein sequence, but the structure is yet to be known, you could use the sequence to what find what the structure could look like and possibly the function.
So how do you find the sequence?
Yes, that is what we call homology modeling. Okay? Homology modeling. Homology is talking about ancestral relationship. Okay, yeah, you model from the ancestry. All right. Okay, you ask where you're going to see the sequence.
I get this.
You don't do like this. Pick it for you. Okay, so that will take us to the basic bioinformatics aspect of this class. Okay, many of us have said about bioinformatics before, right?
Yeah, so
We jumped apart during the listening. Okay, so bioinformatics will um, one of its basic role is as a data repository. Okay, is a kind of an institute or okay, let me say in bioinformatics, it certainly it simply deal with servers that are concerned with data repository and reposition. What it means is storage of data, making data accessible for researchers. Okay, all forms of data, usually genome related data. Okay, anything that has to do with gene sequence, amino acid sequence, protein sequence, structures, and all that. We do that as, in fact, PDB is an extension of bioinformatics. Is that right? Okay, so in this, one of the major servers of bioinformatics, or one of the major um, storage of data or repository of data is the NCBI. Many of us may have heard about that before, right?
yes,
NCBI,
yes.
National Center for Biotechnology Information.
So sometimes if you are downloading something online,
Okay, used to see that, right? Yes, certainly that. Okay, so I will hit on enter just by typing NCBI, National Center for Biotechnology. Just write NCBI, you can investigate that later. Okay, so if you type that, the first thing I will show is this. All right, then you hit on their home page, and then this will appear. There are certainly a whole lot of things you can actually do with this particular thing. Okay, there are a whole lot of things you can do with this particular thing. Many of them, even the ones outside this, maybe at the end of this class, I will share um, some access to some of the classes I have done on this, which have been recorded and uploaded to my channel and all that. Certainly a whole lot you can do here.
you have channel.
Yeah,
Oh, you know about it.
I will know after now. All right. So this is the home page. Okay. Yeah, what I was saying before, one of what you could do here is molecular weight, what they call novel organism identification. You extracted an organism from somewhere here, and then you want to identify what the organism means or is all about. You might not have PCR here, but after sequencing the gene for you, you could work from there. You understand? Yeah, so that's one of the you can you can investigate why they sent, because after that they will send you a name, right? As a scientist, you need to know the name, why they sent you that name, if it's correct and all that. Me, I investigate all that. If it's not correct, I will call you and say, see, there's one of them. I've called them and said, no, no, no,
It's not correct.
it is not correct. He said, please, I should give them a little time, let them that they may have mixed my my own code with someone else, and at the end they attended to that.
And that is if you know.
That is actually if you know.
How can you don't know?
If you don't know, you're on your own. All right. Okay. So this is what we have. Okay. If you open the home page this way, there are many information you can find here. Even PubChem I mentioned before, you can see it as a popular resource attached here. You can see PubMed, um, genome nucleotide, and all that. If you come to this all right, there is all database that is here, right? And then if you click on this arrow, all means anything you search here will respond to all the information in the glucose, right? But if you come here, assuming you're looking for protein, okay, you could come down and then this is protein, all right? If you select protein, when you search item here, what do you think will happen?
Only just
Related to protein, right? Okay, so currently I don't have a particular protein in mind that its structure has not been elucidated, but we will act as if the one we're working with has not. You understand that? What I would have done is to download that and maybe carry the sequence and go and keep somewhere and now come and tell people that this sequence has not been known. You understand? Then we'll investigate the structure, but we could do that actually together. So what do I do? I'll search for that protein. What's that protein we're working with again? Cyclooxygenase 2, right? Cyclooxygenase
I
2.
think you type cost too. Will it come out?
It should. It should. Okay. But most I write the full in case there is something else that is represented as that. Alright. So I will simply hit on the search button. Is that fine? So I'll hit on search and then it will load and then this is what I have. Okay. So it has loaded. It's giving me different cyclooxygenases and their different sources and all that. Alright. Look at the human own, which we might end up working with. That muscus this thing. Yes, yes, yes. It should. If you click more, you have over one page out of 1042 pages. You understand? So, you can click, but let's work with that. Where is that?
How is this faster?
Pasta is talking about the sequence, the amino acid sequence of the protein. Have I passed? Okay, this is the Homo sapien, right? So you can open it in a new tab. All right. Did you see the musculus?
after, before, after.
The human.
No,
Okay.
before this one. So,
Okay. Yeah, I see the muscus. Okay, so we have opened this, right? All right. So this is from human source. If you open that, you're going to find a lot of information with respect to that protein. You just mentioned, of course, it's from human, and then this is the name, this is the ascension number, and all that. If you as well go down, there are many. All these related information here is what you can actually read with respect to that. All right. Including the 3D structure. Of course, he already has the 3D structure, which we know, right? But if he doesn't have, and this was done from X-ray diffraction with the resolution of this, alright? So, but we're assuming we don't have that. Is that fine? Okay, so what we need here is the FASTA. Okay, we're not downloading the PDB structure. We're assuming it doesn't have structure, right? But it will certainly have a sequence. The FASTA is where the sequence is located. If it's organism you're looking at, you're working on, the FASTA will contain the nucleotide bases. Do you understand? While if it's protein, the FASTA will contain the what? The amino acid sequences, right? Amino acid makes up a protein, right? While nucleotide bases makes up a gene. Is that right? Okay, so I'll hit on the faster sequence. Okay, and then if I hit on faster sequence, this is what will appear with the name. All right? Yeah, faster sequence, like I mentioned, is amino acid. They are represented with alphabet. So remember which one?
So this is the sequence.
Yeah, this is the sequence of that. If you count, it's supposed to be 604, which is yes. Now they already told us here. This is 604 amino acid. So the sequence here should correspond to that. Is that alright? So and it's already in short forms for amino acids are written in alphabet.
Yes.
Alright. Yeah. Just like nucleotides as well. Alright. So this is what we have. However, in nucleotide you only have ATCG, right? Adenine, guanine, cytosine, and uracil. You don't know what we're talking about.
Those now.
Bla-
Meditated.
Let me say for myself. I don't know.
Bla, is it true?
Shelter is a fear. Not that you're saying it. Getting me, I remember.
There should be a section of your course where you talk about
Which
it's
department?
even in first year 101 and
Which department?
One
151 is in fact 151. G.
That is first year now. That's why I say that you're saying it. You're trying to remember it. Science lab tech.
but science lab tech chemistry. I beg you differentiate.
Okay, chemistry.
Like somebody in physics that if you're telling this person this thing, they'll be looking at you.
Really?
Like science lab.
Who did biochemistry?
No,
We have SLT
biochemistry. We
biochemistry.
have ICB
Okay.
and we have pharmacology.
Okay. Then are you
Okay.
do biochem?
Okay. It's well. Una don't win. Let us continue.
So your turn.
Alright. So this is actually the sequence. Alright. So I'm going to copy this sequence. Remember, we're assuming we don't know this, right? So I'll copy this and I'll paste it in a notepad just to keep it safe there. I'll paste it here. Okay, this is the sequence, alright? So if I didn't know or this protein doesn't have a structure, I already have a sequence now, right? The next I do now is to go and use it to look for what? A structure. Okay? Looking for a structure is simply the modeling we're talking about.
Okay.
Is that alright? We'll continue. Alright, so we have got our sequence and then this is amino acid sequence, right? For that protein. So the next is to model the protein from this, alright? Okay, so what do? I will return to uh, my internet and then I will check the server we use for such modeling. It's called Swiss model. Yeah, Swiss model. Okay, if you just type Swiss model and then hit on enter, it's um, supposed to show up this way. Alright. So as usual, you click the first option here. Then this will show. You make sure your internet access is good and not like hit up people. Once your internet access is should I hit up people still? Okay.
But sometimes my
I thought she's staying. Alright. So is well. Okay. So this is the homepage, right? You have to start modeling. Okay? Start modeling. If you click on that, this is the homepage. It will show you. In this box, you're going to see where you can paste your target sequence. That is the sequence of the organism or the minor the protein or the uniprot assumption number. Okay. Yeah, uniprot is another server. Okay, I think I mentioned that yesterday. You can use PDB to access your proteins or uniprot. Okay, if you accessed it from uniprot, you might have a server, the ascension number assigned to that sequence. So here we have our sequence. So what do? We go and copy our sequence. This is the sequence, right? So I'll simply copy it.
Go back to notepad.
Yes, where you pasted it, or if it's in the internet where you pasted it there, where you found it, you can as well copy from there. Alright, so I'll paste it. Let's see what will happen when I do the pasting. Alright, so if I paste it, something happened, right? Yeah, once it's recognized as a sequence, it will change coloration, right? It will be colored this way. That's tell you that it was right, the sequence was right. Okay, once you paste it, you color it this way as usual. Then there is option for adding maybe a hetero target. If there's a portion of this you don't want to include in the search, because virtually what you're going to do here is to use this sequence to search the database. That is the modeling. You are using this to search the database and see protein structures that had this similar sequence. All right. Yeah, we'll find out if this will take us to cyclohexygenase or acetylcholine. All right. Assuming we don't know the name of this. So what do I do? I will have to also, if you are in a haze that you might not wait for this to complete, you can include your email address here and title the project. Immediately it's done, it will forward it to your mail. Alright? But we might not have need of that for now. So what do we do? You first of all search for templates. Okay, where you're
We
supposed to
won't do anything. We leave the
email.
this thing.
No, we're not adding any.
So we go to search for template.
Yeah, just go to what? Search for templates. Alright? So I'll click on search for templates, and then depending on your internet stability or strength. Okay, this will go. Um, as you can see that it's trying to search that. Say the search for template matching your target is currently what running. That you should you can see is increasing gradually. Is increasing gradually. So this is what we have. Is running this against this. Is searching for PDB profile database. Is also running blast, right? We know what blast means, right? Blast is just like searching. We call it basic. Gini, gini,
I know.
gini. Birthday blast. The one you know is birthday blast. Okay, blast is like a kind of a holistic search of the database. Okay? That is what blast means. You can see that it's working with over 544 templates. So that might be
How long is this one going to take?
no, it doesn't take time. My system is not like some people.
Is that it?
It's you and Lomi.
It's a me.
Alright, so to soon get completed. So after this, there, like I said, there are many things you could learn with this particular skill. It's the type of skill that don't restrict you to any thing. The more you work with it, the more you will see things to work with it. Is that right? So most of, like I always tell people, most of what I can do just for this little knowledge of this was never taught at the cost of learning this. Alright? The more you work, the more you see challenges and the more you continue working. Is that right? Okay, so this is what we have here. Let's see if um, our search were correct. At the end, it was able to get over 35 templates out of the 500 that it actually extracted information from. Okay, and then at the end, it has sorted it, okay, and then picking about this first one. Do you remember this IKR?
Yes.
Do you remember this
It's for
IKR?
human Homo sapiens. Is it not? Is it not?
Yes, for that sapien. this
That
thing.
article
that you
we opened that day, this was actually I remember that
ID.
the ID where we saw that women even has that. So from our side, this was what they gave us as the number one search.
another humans.
Yes, yes. If we go down, we're supposed to have 3Q something something, right? Um, there 3Q70, right? Except that one is for muscus. Except that is not really.
Everything is like human.
Yeah, because we picked a human this thing. However, if that is not too okay, that might not be very, very close in similarity with what we have here. Alright? So we just have about 35 of that. So what do we do? Normally, before you build a model from this, you um, you consider you just pick say about 5 of them. Okay, the 5 top ones you have here. Meanwhile, what you're seeing here, you can see some blue coverings. You can see a box with a blue covering inside, right? Yeah, in our next page, you're going to understand better what this means. But this simply means the blue is a portion of your sequence against the sequence, the model here. This blue is trying to tell you how similar your sequence was or is to uh, the model you're seeing here, the template you're seeing here. So it means that your sequence was almost the same as.
Who's the one first? Yeah, some I saw that are very small, like
Yes, as you go down, it should be reducing. Okay, it should be reducing. Some are even introducing some white distinct inside it. Okay, if they are quite dissimilar, then you can see this one. Can you see them?
these are not
You
similar.
can see,
The one
yeah, you
very
can even see this one. All right, they are quite very, very uh, low. Okay, so what do? We simply pick the best 5 here. 3, 4. If you notice what is happening here, as I'm adding here, it's adding here, right? Okay, so this is what I have. This is the protein structure. Okay, and of course, it's a cyclooxygenase, which means it actually got it right. As you're hovering around this, you should be seeing your amino acids here. Can you see that? This is chain A and all that. Okay, so I've selected up to 5. I'll have to build what? Model. Alright? So I'll have to build model from that by clicking on build model. And then when I build my model, um, it will start reading this way. By building the model out of the 5, you still tell this to look more closely into that 5
you pick again.
and still pick again. Do you understand? And still pick again the best for me. Think is what is running now, and it's looking like the model 3 is even getting better than the model 1. Model 1 was the top from the other end, but from what you can see, is now the second one here, which means model 3 could be more closer to this. However, we'll analyze that structurally now. So this is the protein, alright? I think this is a double chain though. Yeah, this is a double chain. See the heme group. Do you see that heme group? Then see the ligand. This is not actually naproxen. This is another ligand, right? Okay, Abi is naproxen.
It's not present.
Okay, it's naproxen. Is it naproxen?
Like
It's not. There's up to 3 benzene ring here. However, when you open it up, you see what the substrate there or inhibitor there is. Alright? Okay, so it has finished loading. These are the 5 templates. Okay, let's pick the number 1 and then look at the structural um, analysis or assessment of this particular one and see why this one came as the first one here. The first thing you see here is the GMQE value. This is let me show you, is a quality estimate value. This GME, let me see. meaning of this in protein modeling. Alright. Okay, this is the global model quality estimation. Alright, it's all about the quality estimation. So the value, the closer the value to one, the better. Alright, yeah, it keeps increasing until it gets to one. If it's 100%, then you have one. Then aside that, you have what we call the sequence identity. Alright, you have the sequence identity, which is about 99.82. Alright, which is quite high. However, that first one that was highlighted by us was actually what? 100%. Alright, but I feel we will still find out the reason why this one. Yeah, I don't know if you're getting it. So there should be a reason why this third one, was preferred to that. Alright, so this is the coverage, that's the percentage identity and percentage coverage. Then to read more about the structure, if you look, there's an arrow here. Alright, you'll find an arrow here. So the first arrow will give you what we call the local quality estimate plot or graph. Alright, if you tap on this, it will bolden it. Alright, this is the local quality estimate, trying to tell you how the quality of the model with respect to your protein. Alright, to your model you are looking out for. This is supposed to be downloaded when you are working on modeling. If you click on this image here, okay, you're supposed to uh, enter here as a save. Alright,
So wait, you're supposed to have that this thing, like the local quality estimate.
yes, yes, okay, is one of what you should have in your results. You're going to save this and upload as part of your result. Alright,
That's if you modeled it.
yeah, if you modeled it. Okay, similarly, if we click on the second disk, you're going to find another very important one. I'll click on this. You're going to find another very important one. This is the what they call the z-score value of your model. Okay, the z-score value of your model. As you can see, this particular kind of plot here has three layers. It has a very faint one, it has a little bit deep one, and then it has the deepest one. Did you notice that as well? Are you seeing what I'm doing? You are not wearing eyeglasses.
I'm saying, am I blind?
So there are three of them here, alright? So look at our model. This is the star model, right? Look at the model here. Can you see the model? Look at the model here.
That red thing.
Yes. Look at it here, the red star. If you look, the red star is touching this deep, yeah, trying to tell you the level of or the quality of the model with respect to the template you have. Do you understand? So something like if this star was found somewhere here in this very faint one, and this is not very similar, right? The closer this to this portion, the higher. So this closeness as well has a score here. If the z score is greater than 2, it means it's in the faint one, right? If the z score is less than 2 but greater than 1, it's still in the average, the center one. However, if it's less than 1, it means it's in the inner one or the deepest one. So in your abstract, you write something like a model with a z score of less than 1. We already know that this was actually in the center, in the deepest portion. Alright? Okay, so that is what we have here. So this is also supposed to be saved by clicking on that. Is that alright? We call that quality comparison plot. Okay, so this being done, we go ahead to take the structural assessment of this protein as well, or this model. Alright? If you click on structural assessment, it will load and it will give you another important graph again. There are not many graphs, right? Yeah, we'll call this the Ramachandran plot. If you start reading works where modeling was done, you'll find out a Ramachandran plot as well. All these small this thing, this is the protein. And then, as usual, if you look at this Ramachandran plot, you discover that this is our gain function for some of us that did quantum chemistry. We've gone to chemistry and we've come across this before, right? Alright, so this is the plot. Yeah, if you know Ramachandran plot, these are the coordinates and all that. So in this plot as well, there are three major um, layers here. You have the very faint, you have the a little bit deep, and you have the deepest green, right? The locality or location of these are the amino acids. If I'm touching this, you should be seeing it here.
Okay.
Alright? Yes, as I'm touching, you should be seeing it here and all that. So the ability of these amino acids to be located in the inner portion, okay, also tells you how quality, yeah, the template or the model you're working from now is. If these are all distributed outside here, like you can see few of them, it's trying to tell you they are not really similar.
No, let me see some.
Can you see that? Are you seeing it?
Yes.
Yes, if you turn this, it should be somewhere.
Here is the heme group again.
In group is there. See
This one.
yes.
Now look like ladder.
Yes. Look at it. Look at it.
Oh,
It
this
should be two. Another one should be. There's one that is here. There's one that's supposed to be here. In this model, only one is here. I understand. Okay, so this is the what? The plot. As well, you click on this to download that as well, right? There's a camera here and all that. So there's what we call Ramachandran favored and outlier. Okay, you discover that this has about 99.9% Ramachandran favored, and then an outlier of about 0.00. Okay, meaning this has actually a good quality. All right. Yeah, all these some people will publish, and then all these are what they will populate the write up with, which are actually necessary and all that. All right. So if that is being taken, we can now go back to this is model 3. And now go back to um, our structure and then pick our model. Where is our model 3? Okay, this is our model 3. I've clicked and returned back, so it has tried to reshuffle as if I didn't agree with what it gave me before. Okay, so this is the model 3 on. If you come to the portion here, we've seen this before, right? If you come to this, you see download file. Alright, so if you click on download file, you can download this as PDB format. You can download it as other format. Here we're interested in what the PDB format. So I can click on the PDB, then it has downloaded. Okay, I can open that by clicking on that, and then it will load the Discovery Studio, and then open the protein we just downloaded. Alright, so this is the protein just downloaded. Can you see that? This is the protein, uh, very similar to what we had earlier. Okay, this is chain B, this is chain A. Alright, and then it also has heat atoms as well. Now highlighted. Alright, so with this, we've been able to get a 3D structure of a protein to work with, right? And then we can now continue our docking process from here.
So you clean.
Yeah, you can now start cleaning, docking, and all that. Alright, so this um, is the portion or the remaining aspect of that in case you run into such challenge in the nearest future. Now you have handled when you don't have your compound in the database, right? And as well, when you don't have your protein in the what in the database, is that fine? So, and then how you can go about them if you don't have any of this. So these are the only few major challenges you could encounter in the process of doing this, which I did then, and I thought it wise to explain to those that might go into that as well. So the remaining part of the task is now on you guys' shoulder. Immediately after this, I'm going to give everyone a project here to execute immediately. Some people, yes, some people, no, you won't do it here. So that your mind will not feel like you will go home, study this. This recording will be shared to us uh, after this assignment I gave us and it has been submitted. Hmm? Mhm. I'll now share these recordings to us, and then we could build more from that, and then work on the project and bring to give to us. Is that fine?
My
Yes.
project should be on my list, my